But give me meters. I don't really know how long now.
Hello guys, we are back with another video. Hope you have watched the previous videos and solved your queries. Please subscribe our channel and press the bell icon. Subscribe us so we can grow together. Also follow us on Facebook, Instagram and TikTok. Links are given in the description and the name is Dr. Buzz Lecture. Today's lecture is glycogenolysis, which is also known as glycogen metabolism. Glycogen is basically a polymer which is made up of glucose chain and which is held by glycosidic bond, which can be 1,4 glycosidic bond or in linear manner or 1,6 glycosidic bond in branch manner. When we eat food, the glucose level in the body goes high, which causes pancreas to release insulin. When insulin is released, glycolysis happens and we create more ATP. When there is more production of ATP, it inhibits glycolysis because there is no need of more ATP. Then to use more glucose in other processes, glycogen metabolism happens. There are three steps of glycogen metabolism. First is attaching uridine disphosphate to glucose. Second is attaching this glucose part to the glycogen which works as a primer. And third is adding branches. So let's see how these steps work on. As you can see, first step is attaching UDP to glucose. In this process, as we know, first glucose is converted into glucose 6-phosphate by the enzyme called hexokinase. Then glycogen metabolism happens. Glucose 6-phosphate then converted into glucose 1-phosphate from the enzyme called phosphoglucomutase. Then glucose 1-phosphate is converted into UDP glucose, in which UDP is attached with glucose. As UDP is a diphosphate, so 1-phosphate comes from a uridine triphosphate, from which 2-phosphate molecule is used in energy and 1-phosphate molecule which is left is used to make UDP glucose. The second step of glycogen metabolism is attaching glucose part of UDP to glycogen. As we know that glycogen synthase adds more glucose to the glucose chain, which should be at least 4 molecule long. If it is not 4 molecule long, then glycogen takes part and works as a primer and fools glycogen synthase. And this causes glucose add in the glucose chain. The second step is not that big, it consists of only one step, an addition of glucose. In it basically would detach UDP from glucose. UDP is detached from glucose and glucose molecule is free. Here glycogen can take a part if the chain is not 4 molecule long, it works as a primer. So when UDP will detach, the glucose molecule will go to the glucose chain where glycogen synthase will add it into the chain. So likewise more glucose will be added in it. The third step is called adding branches, in which at start we add glucose in linear manner in the 1-4 glycosidic bond. So when more glucose wants to add up in glucose chain, then the glucose chain will change its shape from linear manner to branch. For this, 1-4 glycosidic bond will be cut off and the glucose chain will arrange in branch manner by making 1,6 glycosidic bond. So this was the last step of glycogen metabolism. As we know that when glucose molecule is high in the body, first glycolysis happen. If ATP is achieved, then glycogen metabolism happen. What if when there is low blood glucose level in the body when we are exercising, then glucagon will be released in the body, which will cause liver for the breakdown of glycogen. Moreover, epinephrine can also cause skeletal muscle for the breakdown of glycogen. Basically, there are two types of enzymes which take part in breakdown of glycogen. First one is glycogen phosphorylase. The function of glycogen phosphorylase is that it adds up phosphate ion into the glucose molecule and convert into glucose 1-phosphate. A second enzyme which take part in the glycogen breakdown is debranching enzymes. It acts on branching part of glucose chain. The first enzyme is 4-alpha glucanotransferase, which shifts 3 glucose molecules from branch chain to the linear chain. The second enzyme which takes part is alpha-1,6-glucosidase, which deducts one remaining branching glucose molecules and it makes it free to move. So here we get one glucose molecule. Secondly, we also have to discuss about glucose 1-phosphate, which can be converted back into glucose. Glucose 1-phosphate then can be converted back into glucose 6-phosphate because it is a reversible process. So same enzyme will take part, which is phosphoglucomutase. Then glucose 6-phosphate is that again converted into glucose by hexokinase. That's how glycogen breakdown works. Now let's see how these processes are stimulated within the cell. Glycogen synthesis happens when there is more glucose in the body. So when there is more glucose, more insulin will be released. Insulin will stimulate a receptor which is present inside the cell membrane, which is called tyrosine kinase receptor. The function of tyrosine kinase receptor is to stimulate protein phosphatase, which will remove phosphate and which will activate glycogen synthase and which will inactivate glycogen phosphorylase. Moreover, when in glycogen breakdown, when we need more glucose, then glucagon stimulates G protein coupled receptor inside the cell membrane. G protein coupled receptors stimulate adenylyl cyclase. With the help of ATP, it will convert into CAMP and which stimulate kinase A to add phosphate. By adding phosphate, it activates glycogen phosphorylase and inactivates glycogen synthase. So the two main components which carry on glycogenolysis is glycogen synthase and glycogen phosphorylase. Thanks for watching the video. Hope you liked it and you have solved all your queries regarding the topic. Please subscribe our channel and if you have any query, please comment us. I will come back with another new video. Till then, bye bye.
But give me readers. I don't really